There are two groups of prokaryotic cells, the bacteria and the archaea. Prokaryotic cells are different from eukaryotic cells because they have a cell wall, cytoplasm, and ribosomes, unlike a eukaryotic cell which has a nucleus and other organelles. Fundamentally, two different types of cells exist. Prokaryotic cells are so named because they lack a membrane-bounded nucleus. The other type of cells, called eukaryotic cells, has a nucleus. Prokaryotes are present in great numbers in the air, in bodies of water, in the soil, and they also live in and on other organisms. Although they are structurally less complicated than eukaryotes, their metabolic capabilities as a group far exceed those of eukaryotes. Prokaryotes are an extremely successful group of organisms whose evolutionary history date back to the first cells on Earth. Prokaryotic cells are divided into two types, largely based on DNA and RNA-based sequence differences. The two groups of prokaryotes are so biochemically different that they have been placed in separate domains, called domain bacteria and domain archaea. Bacteria are well known because they cause some serious diseases, such as tuberculosis, anthrax, tetanus, throat infections, and gonorrhea. But many species of bacteria are important to the environment because they decompose the remains of dead organisms and contribute to ecological cycles. Bacteria also assist humans in still other ways. We use them to manufacture all sorts of products, from industrial chemicals of foodstuffs and drugs. For example, today we know how to place human genes in bacteria so that they will produce human insulin, a necessary hormone for the treatment of diabetes.
Prokaryotes are quite small. An average size is 1.1 to 1.5 wide and 2.0 to 6.0 long. While other prokaryote shapes have been identified, three basic shapes are most common. A rod-shaped bacterium is called bacillus, while spherical-shaped bacterium is called coccus. Both of these occur as pairs or chains, and in addition, cocci can occur in clusters. Some long rods are twisted into spirals, in which case they are spirilla. If they get rigid, or spirochetes, if they are 4.4 shows generalized structure of bacterium. This means not all bacteria have the structures depicted, and some have more than one each. Also, for the sake of discussion, we will divide the organization of bacteria into the cell envelope, the cytoplasm, and the appendages.
The cell envelope includes the plasma membrane, the cell wall, the glycocalyx. The plasma membrane is a phospholipid bilayer with embedded proteins. The plasma membrane has the important function of regulating the entrance and exit of substances in and out of the cytoplasm. After all, the cytoplasm has a normal composition that needs to be maintained. In prokaryotes, the plasma membrane can form internal pouches called mesosomes. Mesosomes most likely increase the internal surface area for the attachment of enzymes that are carrying on metabolic activity. The cell wall, when present, maintains the shape of the cell. Even the cytoplasm should happen to take up an abundance of water. You may recall that the cell wall of a plant cell is strengthened by the presence of cellulose, while the cell wall of bacterium contains peptidoglycan, a complex molecule containing a unique amino disaccharide and peptide fragments. The glycocalyx is a layer of polysaccharides lying outside the cell wall in some bacteria. While the layer is well organized and not easily washed off, it is called a capsule. A slime layer, on the other hand, is not well organized and is easily removed. The glycocalyx aids against drying out and helps bacteria resist a host's immune system. It also helps bacteria attach to most any surface.
The cytoplasm is a semi-fluid solution composed of water and inorganic and organic molecules, encased by a plasma membrane. Among the organic molecules are a variety of enzymes which speed the many types of chemical reactions involved in metabolism. The DNA of a prokaryote is found in a chromosome that coils up and is located in a region called the nucleus. Many bacteria also have extra chromosomal piece, circular DNA called plasmid. Plasmids are routinely used in biotechnology laboratories as vectors to transport DNA into bacterium. Even human DNA can be put into bacterium by using a plasmid as a vector. This technology is important in the production of new medicines. The many proteins specified for bacterial DNA are synthesized on tiny particles called ribosomes. A bacterial cell contains thousands of ribosomes that are smaller than eukaryote ribosomes. However, bacterial ribosomes still contain RNA and protein in two subunits, as do eukaryote ribosomes. The inclusion bodies found in the cytoplasm are stored granules of various substances. Most bacteria metabolize in the same manner as animals, but the cyanobacteria are bacteria that photosynthesize in the same manner as plants. These organisms live in water or in ditches, on buildings, and on the bark of trees. The cytoplasm contains extensive internal membranes called thylakoids, where chlorophyll and other pigments absorb solar energy for the production of carbohydrates. Cyanobacteria, the blue-green bacteria, because some have pigment that adds a shade of blue to the cell, in addition to the green color of chlorophyll. The cyanobacteria release oxygen as a side product of photosynthesis, and perhaps ancestral cyanobacteria were the first types of organisms on Earth to do so. The addition of oxygen changed the composition of the Earth's atmosphere.
The appendages of bacterium, namely the flagella, fimbriae, and conjunction pili, are made of protein. Motile bacteria can propel themselves in water by the means of appendages called flagella. The bacterial flagellum has a filament, a hook, and a basal body. The basal body is a series of rings anchored in the cell wall and membrane. The hook rotates 360 degrees within the basal body, and this motion propels bacteria. The bacterial flagellum does not move back and forth like a whip. Sometimes flagella occur only at two ends of a cell, and sometimes they are dispersed randomly over the surface. The number of location of flagella can be used to help distinguish different types of bacteria. Fimbriae are small bristle-like fibers that sprout from the cell surface. They are not involved in locomotion. Instead, fimbriae attach bacteria to a surface. Conjugation pili are rigid tubular structures used by bacteria to pass DNA from cell to cell. Bacteria reproduce asexually by binary fission, but they can exchange DNA by the way of conjunction pili. They can also take up DNA from the external medium or by way of bacterius.